Introgression is genetic transfer between species. Being able to transfer genetic material from one species to another is done through hybridization. Taking genetic material from one species, from another, and one species and another is known as backcrossing, and this is what helps you build a DNA sequence for the organism that you're wanting to create. Now, there are several mediums or ways that you can hybridize genetics or fuse the DNA together of different organisms, and one of them is known as soniporation, which uses sound. With the use of ultrasonic waves or acoustic microstreaming, this affects the speed at which these microbubbles vibrate at. When these bubbles begin to oscillate, they create push and pulls that make openings or pore formations where genetic material can be inserted and taken out of DNA sequences or cells. Another type of hybridization or a way to introgress genes is known as magnetofection. These magnetic nanoparticles are affected by an external magnetic field, which allow push and pulls for openings and pore formations. And again, those openings allow you to insert genetic material or to take out genetic material. Now, the one that most people are familiar with, especially if you're familiar with those conversations about splicing and cloning, is electroporation. And if you're used to the full length discussion, then you've definitely seen me use this picture before. And actually what was taking place is the biologist Pablo Ross was actually fusing his DNA with a pig's. And the medium of hybridization that he used for that was electroporation. And this is Pablo Ross, by the way. Now, when it comes to introgression and taking the DNA material that you want to fuse together, scientists have created a centralized way to do so. So the CRISPR part of this tech particularly deals with the bacterium. The Cas9 or protein 9 is what actually cuts and allows for genetic transfer. Now, one of the driving forces behind CRISPR-Cas9 is Miss Jennifer Doudna. One of the things that I find interesting about her interviews is how she explains that by the time that she actually got her hands involved with this tech, that it was already widely being used by the world. So I don't know why she's really the face of something that's already been widely used. I mean, we already know that dogs and cats are genetically modified organisms. Broccoli and kale are genetically modified organisms. So y'all were splicing, or there was an entity or some type of splicing that was going on for a very long time. They said that dogs were first domesticated 15,000+ years ago. So if y'all are talking about how y'all got y'all's hands on it today, explain to me how a dog has introgress genes and it was first domesticated 15,000 years ago. That's a conversation for another day. Now, CRISPR became widely used in 2020, and to be honest, it's old news. There is now a new gene editing tool known as Stitcher. And see, the difference between Stitcher and CRISPR is that CRISPR only allowed them to match a desired target gene. It only allowed them to cause double-strand DNA breaks or, modifications to the genome. But Stitcher is like CRISPR on steroids. This allows you to insert and modify an entire gene. This tool offers a one-and-done approach that overcomes hurdles from CRISPR gene editing tech. And it goes on to say which programmed correct individual mutations. So it says Stitcher allows the harnessing of different enzymes from genetic elements called retrotransposons, which are found in all eukaryotic cells. Y'all know I ain't gonna let that go, including animals, fungi, and plants. They are often called jumping genes for their tendency to move around and insert themselves into the genome. But look, this is how they're using it. The researchers recognize how to copy and paste that mechanism they could use to move and to repurpose and edit genes at specific locations. New gene editing tool shows promise for treating diseases. Where do all venereal diseases come from? I just want to see what you're going to say. Who has the genes that mutate? I just want to see what you're going to say. There's a fine line between birth defects and gene mutations. That's a conversation for another day. Now, we're going to keep talking more and more about introgression, hybridization, how they're using it, when it first started, how they had to start small. Because what people fail to realize is that how were they able to get to the point of CRISPR-Cas9? In order for you to be able to do this, you have to start small. And we're going to really pay attention and hone in on how they started small and how they worked their way past CRISPR into Stitcher.